Chapter 5, The Working Cell. We've learned about cells in the previous chapter. A cell is a basic unit of life. You can imagine the cell as a machine that works continuously and efficiently to perform a wide variety of functions such as movement, processing of cellular energy, and also production or secretion of different products. In this chapter, we will explore three processes common to all living cells. First, energy metabolism. Second, the use of enzymes to speed chemical reactions. And third is the transport regulation by the plasma membrane. Along the way, we'll further consider nanotechnologies that mimic the natural activities of living cells. Even the smallest cell, such as this human white blood cell, is a very complex miniature machine. Energy concepts. What is energy? Energy can be defined as the capacity to cause change. It is the ability to work. And there are different forms of energy. The two forms of energy are kinetic energy and potential energy. Kinetic energy means the energy of motion, while potential energy is whatever energy is stored in an object because of where it is located or because of its structure. And energy just converts from one form to another. There is a physical principle which is called the conservation of energy, and it states that energy can neither be created nor destroyed. It can simply be converted from one form to another. In other words, the total energy is conserved. Every energy transformation releases some randomized energy in the form of heat. Heat is a type of kinetic energy contained in the random motion of atoms and molecules, and heat has the highest entropy. Entropy means a measure of disorder or randomness in a system. Every time energy is converted from one form to another, entropy would increase. Molecules store varying amounts of potential energy in the arrangement of their atoms. Organic compounds are relatively rich in such chemical energy. The combustion of gasoline within a car's engine and the breakdown of glucose by cellular respiration within living cells are both examples of how the chemical energy stored in molecules can be converted to useful work. Let's try to understand the energy transformations during a dive. To get to the top of the platform, the diver has to convert the chemical energy obtained from food into kinetic energy of the muscular movement required for climbing. At the platform, the kinetic energy of the muscle movement is converted into potential energy because of the elevated location. Then the act of diving off the platform into the water converts the potential energy back to kinetic energy. Cellular respiration is the process by which glucose and other fuel molecules are broken down to release energy, and that energy is captured and kept in the cell as ATP, adenosine triphosphate, which is a form that the cell can use to perform work. Food calories, actually kilocalories, are units used to measure the amount of energy in our foods and the amount of energy we expend in various activities. A calorie is the amount of energy that can raise the temperature of 1 gram of water by 1 degree Celsius.
Energy is the capacity to cause change. Kinetic energy is the energy associated with motion. Energy that is not kinetic is called potential energy, and is energy that matter possesses because of its location or structure. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy.
The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow. When the arrow hits the target, its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let's rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, some of the energy can be used to do work, but some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. As the arrow flies through the air, heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Heat energy is a very disordered kind of energy. It has the highest amount of entropy, or disorder, of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. When your body breaks down food molecules, the stored potential energy from food can be converted to kinetic energy. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP. Water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work, such as pulling back a bowstring, but the heat energy that is generated cannot be used to do work.
ATP stands for adenosine triphosphate. This is the energy that is generated by the cell in the mitochondria. This is the molecular currency and the type of energy that the cell can use to perform various cellular functions. Your cells recycle ATP. As ATP is broken down to ADP to drive cellular work, new molecules of ATP are built from ADP using energy obtained from food. ATP is the source of energy because ATP energizes other molecules by transferring a chemical group called a phosphate group to those molecules. How ATP drives cellular work. Each type of work shown here is powered when an enzyme transfers phosphate from ATP to a recipient molecule. The ATP cycle basically explains that energy from processes that yield energy, such as the breakdown of organic fuels, is transferred to processes that consume energy, such as muscle contraction and other cellular work. Metabolism is defined as the sum total of all chemical reactions occurring inside an organism. Metabolism is not possible in the absence of enzymes. What are enzymes? Enzymes are a group of proteins that speed up or enhance chemical reactions in biological systems without themselves getting consumed in the reaction. All living cells, they contain thousands of different types of these proteins or enzymes. Enzymes act as biological catalysts that speed up metabolic chemical reactions by lowering the activation energy required to break the bonds of reactant molecules and triggering a chemical reaction. This figure illustrates how the activation energy barrier is reduced by enzymes. Can enzymes be engineered? Like all proteins, enzymes are encoded by genes, and genetic sequences suggest that many of our genes were formed through a type of molecular evolution. A technique called directed evolution was used to produce a better version of a particular enzyme. Scientists mutated many copies of a gene that codes for a particular enzyme. The genes for the enzymes that were most effective were kept, then subjected to more rounds of duplication, mutation, and screening. After many rounds, scientists were able to isolate a gene that codes for an enzyme which is 170 times more efficient at promoting the reaction than the original enzyme. This figure illustrates the directed evolution of the enzyme glycoside hydrolase, which can remove compatibility proteins from the outside of red blood cells. Enzymatic activity. Enzymes are very selective in the reaction that they can catalyze. And this is because each enzyme can recognize only a specific reactant molecule, which is known as a substrate. There is a site on the enzyme that is complementary in shape and chemistry to that of the substrate, and that particular site is known as active site, and this interaction is called induced fit. So the entry of a substrate into the active site of an enzyme causes the enzyme to change shape slightly, allowing for a better fit and thereby promoting the interaction of enzyme with substrate. After the reaction products are released from the active site, the enzyme is free to accept another molecule of its substrate. So this ability for enzymes to be reused is a key function of enzymatic activity. This figure depicts how the enzyme lactase works, and the substrate for this enzyme is lactose. Enzyme inhibitors are molecules that can disrupt metabolic reactions by binding to the enzymes either at the active site as substrate imposters that plug up the active site or elsewhere. This figure shows how enzyme inhibitors stop metabolic reactions by not letting the enzymes work. Plasma membrane function. Remember, we learned that cells are separated from their external environment because of the presence of a plasma membrane or a cell membrane, and the cell membrane is made up of a phospholipid bilayer. That means two layers of fat that has proteins embedded in it. The figure shows the phospholipid bilayer of the plasma membrane and the various membrane proteins there with their functions. There are two types of transport that occur across a plasma membrane: passive transport and active transport. The difference between the two types of transport is the requirement of ATP. For passive transport, the cells don't need to spend any ATP. For active transport, cells need to spend ATP. That means ATP is broken down to release energy. Now, the first type of passive transport is diffusion. Diffusion means the movement of molecules from a region of high concentration to a region of low concentration across or down a concentration gradient. That means at two places there is a difference in the concentrations, and then when molecules move from that place which has a higher concentration of that particular molecule to another place which has lower concentration, then that particular movement would be known as diffusion. This figure illustrates diffusion across membranes. A second type of passive transport is called facilitated diffusion, in which molecules can diffuse but they need some help, and that help comes from the proteins of the plasma membranes, which act as tunnels or corridors for specific molecules, and those molecules can pass through these corridors. The third type of passive transport is called osmosis. Osmosis is defined as the diffusion of water molecules across a concentration gradient from a region of high water concentration to a region of low water concentration across a selectively permeable membrane. Since osmosis is movement of water molecules, this movement would be in the direction that is opposite to the movement of solute molecules. So if you are considering a solution that has glucose or sugar dissolved in water, and you have two different concentrations of the sugar solution here, the part where there is more sugar would have less water, and the other solution which has less sugar has more water. So that means sugar will be moving from, let's say, this particular area where it has more green dots, that means more sugar, whereas water would be moving in the opposite direction. So diffusion of sugar molecule is occurring in one direction, and osmosis of water molecules is moving in the is happening or occurring in the opposite direction. Now there are three definitions here on this slide that you have to memorize and understand: hypertonic, hypotonic, and isotonic. Hypertonic means compared to another solution, this particular solution has a higher concentration of the solute. A hypotonic solution, on the other hand, has a lower concentration of the solute, while an isotonic solution has an equal concentration of solute. Water balance is very important because the survival of a cell depends on its ability to effectively balance water uptake and loss. If an animal or a cell is placed in a hypotonic or hypertonic environment, for this cell to survive, the animal must have a way to balance this water uptake and loss, and this control is known as osmoregulation. Animal cells such as a red blood cell and plant cells behave differently in different osmotic environments. Most animal cells require an isotonic environment with equal concentrations of water within and outside the cell. On the other hand, plant cells need a hypotonic environment which causes water to flow inward, keeping the walled cells turgid. When an animal cell is immersed in an isotonic solution, the cell's volume remains constant because the cell gains water at the same rate that it loses water. But what happens if an animal cell is in contact with a hypotonic solution which has a lower solute concentration than the cell? Due to osmosis, the cell would gain water, swell, and would burst. That means there would be cell lysis, like an overfilled water balloon. A hypertonic environment is also harsh on an animal cell. The cell shrivels from water loss. If a plant cell is immersed in an isotonic solution, it becomes flaccid, that means floppy, and the plant wilts. In contrast, a plant cell is turgid, that means very firm and healthiest in a hypotonic environment with a net inflow of water. Although the elastic cell wall expands a bit, the back pressure it exerts prevents the cell from taking in too much water and bursting. In a hypertonic environment, a plant cell loses water, it shrivels, and its plasma membrane pulls away from the cell wall. This is known as plasmolysis. This usually kills the cell. Because of their rigid cellulose containing cell walls, plant cells are healthiest when they are placed in hypotonic environment with a net inflow of water which expands their cell walls without bursting. Turgor is necessary for plants to retain their upright posture and the extended state of their leaves. Watering a wilted plant will make it regain its turgor. Active transport means a transport across the cell membrane where a cell has to use ATP, break down ATP to release energy. And this transport is usually required when a cell has to move a protein or a certain molecule against the concentration gradient. That means from a region of low concentration to a region of high concentration. This figure shows the three types of passive transport: diffusion, facilitated diffusion, and osmosis, and compares it to active transport. Exocytosis and endocytosis are a special type of active transport known as vesicular transport. Vesicular because it uses vesicles, secretory or transport vesicles. There are two types of vesicular transport. Exo means exit and endo means entry. So exocytosis is the movement of materials out of the cytoplasm of a cell, whereas endocytosis is when a cell takes in a material from outside. So endocytosis means where cell is taking in is further divided into whether the cell is drinking or whether the cell is eating. So if the cell is drinking, that is known as pinocytosis. Remember pina colada. While phagocytosis means a cell is eating. That means the cell is taking in a solid molecule. In pinocytosis, the cell was taking in a liquid molecule or a really small solid molecule. This figure illustrates exocytosis, where a particular particle had to be ejected out or kicked out of the cell using vesicles. This figure shows endocytosis, that means cell drinking or cell eating.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, The sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer.
The spontaneous formation of membranes would have been a critical step in the evolution of first cells.